So it's my pleasure to introduce Dr. Brian Clark, who is a professor of physiology and neuroscience in the Department of Biomedical Sciences at Ohio University. And he is also the Harold E. Clybourne Endowed Research Chair. He is the executive director of the Ohio Musculoskeletal and Neurological Institute, with the abbreviation of OMNI. And it's also my pleasure to introduce him because Dr. Clark was a student of mine years ago. So we just had a nice conversation before this to catch up a little bit. So he did his bachelor's at Western Carolina University in biology, and then he went to Syracuse University for his master's and PhD in exercise physiology. And he also got a certificate of advanced study in gerontology, which you'll see comes into his work. As I said, he is the executive director of OMNI. Since he's been there, I think he got the position around 2008. He's increased the publication rate 6-fold and increased funding approximately 20-fold. So they've been very, very successful with what has been created. He's published more than 150 articles that have been extensively cited. He has received more than 20 million in grant funding. Currently, he has 2 R01s and an R44 and an NMD farm grant, and he has another grant pending. So he's very successful with grant funding. His honors include Presidential Research Scholar at Ohio University in 2018, the J.O. Watson Memorial Lecture Award from the Osteopathic Heritage Foundation in 2017, and he was actually at ACSM a new investigator. He's been on numerous expert boards, councils, task force, NIH study sections. He's been a consultant for Zev Industry and Abbott Labs. He is I think he was one of the original creators and now scientific director for a med tech startup for OsteoDex Incorporated. So he is keeps himself very, very busy. The overall goal of his research is to develop effective and implementable interventions that increase muscle function, muscle strength, motor control, fatigue resistance, and mobility in older adults and/or patients with orthopedic and neurologic disabilities. And back in the day, he was an awesome runner too. So I would like to introduce to you Dr. Clark, and I'll let him go ahead.
Okay, you guys can hear me, correct?
Yes.
Okay, very good. Jill, thanks for the introduction. I was thinking back, I think it's been 17 years now since I was your student, and it seems like it was yesterday at times. So it's sometimes it's staggering to think about how quickly time flies, to say the least. But thanks for giving me a chance to talk to you guys. What I'm planning to speak about today is the neural mechanisms of age-related weakness. It's in large part been the central focus of my work for the past 15 or 16 years. I started out when I was a grad student at Syracuse, primarily studying the neuromuscular mechanisms of disuse-induced loss of muscle strength and muscle function, motor function, broadly speaking. And over the years, I've gradually and not so gradually at times shifted to aging, in large part because of the societal relevance as well as the funding opportunities that have surrounded that. I just always start with my conflicts. As Jill sort of alluded to, I always say I'm a very conflicted person. Particularly, I have worked with a fair number of pharmaceutical companies, and I will be presenting some set of data today that is from at least one of these pharma companies. So that's just, the good housekeeping conflict of interest disclosure. The OsteoDX, that is not related to what I'm presenting today. That's a company that we founded that's trying to develop technologies to assess the mechanical properties of bones in humans, sort of a surrogate to DEXA. But I didn't want to point out the pharma connection. So why study aging? This is a figure from a few years ago that Matt Keberline out of University of Washington put out that I now love to use. And it sort of shows that slowing aging is arguably more effective than curing disease. And it's sort of showing that if you look at the calculated impacts on life expectancy for a typical 50-year-old woman from curing cancer, from curing heart disease, or combined, that slowing aging actually has a much larger extent on extending the life expectancy. The green is showing no comorbidities, and the red are sort of representing individuals with severe comorbidities. So I always start with this, particularly I tend to give a lot of talks to neuroscience programs and neurology groups, and sometimes they wonder, why aren't you studying aging? Why aren't you studying Parkinson's disease or some overt neurological condition? So I always sort of try to set the stage for aging from the big picture. And then, of course, there's this sort of societal shift that's happening within our population. I sometimes refer to it as the graying of the nation. This is just showing the US population projections by age through 2060. And I like the waterfall graph that sort of shows that the proportion of older adults is going to be gradually increasing. And with this comes profound individual and societal burdens. A lot of my talk is going to focus on weakness. So here I want to point out that 40% of older adults have one or more physical limitations in daily tasks that are essential for independence. 30% of older women and 15% of older men report that they cannot lift or carry 10 pounds. Low physical function is one of the most common reasons for needing long-term care, with the total spending projected to be $346 billion. That's a staggering number. And to put that in perspective, I don't know what the annual budget for NIH this year is, but generally I think NIH's budget comes in somewhere between 40 or 45 billion. So when you think about 346 billion for long-term care relative to the entire NIH budget, and I may be off by 10 billion for NIH. I know 15 years ago it was 30 billion. I don't know what it is today, but any way you slice it, that's a ginormous number. Just to kind of, this is sort of showing the disability prevalence across the United States. Nearly 40% have at least one disability, and mobility disability is the most common disability among Americans, with 2/3 of those that have a disability. It's actually a mobility disability, which is commonly defined as simply as you cannot walk a quarter of a mile. I say all of that to try to make the case that age-related weakness, muscle weakness, is a major contributor to disability. Weakness has been shown to predict disability as well as mobility limitations. Its midlife hand grip strength has even been shown at midlife to be a predictor of disability in older age. Weakness has also been shown to predict death or survival. I have this one study here that noted a 6-year longitudinal investigation of 142,000 older adults revealed that low grip strength is a stronger predictor of all-cause mortality than systolic blood pressure. That statistic sort of staggers a lot of folks. I use it in my grants all the time to try to set that hook for why we should care about it. And then recently we published a paper showing that we did some cohort matching, and made the argument that it had a causal association with early mortality. And here you're seeing, sort of a survival curve showing that weakness, people who were weak had a much greater propensity for death in subsequent years. It's also associated with more markers of frailty than chronological age. And that's led a number of people over the years to suggest that weakness should be considered a vital sign of aging, along with gait speed. There's been some great articles advocating that every geriatrics clinic should be taking grip strength as well as gait speed, a 4-meter gait speed or something simple for everybody that comes in. Unfortunately, very few are doing it. I've tried to convince our local geriatrics clinics here at Ohio to do that, and I fall on my face every time because of time. Physicians are already overstretched as well as their staff, and so I've not been successful, but it's something, in case there's any geriatricians on this call, that I would continue to advocate for. And so what I'm going to spend much of my talk talking about is weakness and the mechanisms, but I want to step back for a second and talk a little bit about sarcopenia. I'm sure everybody on this call has heard of sarcopenia. Sarcopenia was originally sort of operationally defined in the late 80s, early 90s as the age-related loss of muscle mass. And then in the mid-90s, that sort of gradually shifted to the age-related loss of muscle mass and strength. And the supposition was that as we get older, we have muscle wasting or atrophy, and that wasting is what was causing weakness, and then that this weakness was driving forward to create mobility limitations. And that was not by any means an illogical assumption at the time, but there's been a lot of data come out over the years suggesting that this is not necessarily a causal association, that weakness is very multifactorial in its mechanisms. And I think I would argue that for those of you who are not in the aging space, there have been a large number of clinical trials of anabolics over the past 15 years. The majority of the most common one that people are familiar with are the myostatin inhibitors. Virtually all of the myostatin inhibitor trials have shown very weak results. They've not shown super promising results, as well as other anabolics. Even though many of them have shown to have some, modest to moderate enhancements in muscle wasting, it's not translated to enhancements in physical function and mobility. And at least to date, the FDA has made it apparently clear that the only way they're going to improve any sort of a compound for age-related physical function enhancement or sort of performance enhancement is that if it actually enhances physical function and mobility. And so it's been a major investment on the pharmaceutical end that's not panned out. And for those who are interested in this, in about 2 or 3 weeks, the NIH is hosting a whole 3-day series of workshops on developing of compounds for promoting function in older adults. You can go on the NIA website and find that information. It's going to be a virtual meeting during which this talk, as well as a series of other ones, will be given. So I'm going to argue that muscle wasting is not the primary cause of muscle weakness, and that this sort of typical causal assumption does not give sufficient consideration to muscle function being a byproduct of motor function. I always say that muscles are to a large extent puppets of the nervous system. And again, I think this likely explains why these anabolic therapies have not translated into functional enhancements. And I want to point out that 15, 16 years ago, myself and actually one of Jill's former students as well, Todd Manini, who I think may have given a talk to you guys a few years ago, because I know Todd told me he had talked to Jill. We wrote a paper arguing that we needed to operationally redefine sarcopenia, and that we needed to keep sarcopenia as the age-related loss of mass, and that we needed to define weakness as dynapenia. And that's been cited a lot, and it's certainly, I think, had the impact that we hoped it would have. But what I want to point out is that just this past year, the 2 most common consensus definitions of sarcopenia actually changed their definition. And the most recent definition of sarcopenia now places weakness as the key characteristic of sarcopenia. So to some extent, I think what Todd and I were arguing should be dynapenia. They've just sort of flipped the definition a bit. And I think one of the reasons, I may not have it in here, I don't think I have it in this particular talk. I think one of the reasons that really pushed this is there's been a series of large experiments and analyses over the past few years looking at the role of weakness predicting mobility issues, as well as the role of low mass, with the biggest one probably being the series of papers from the Sarcopenia Definitions and Outcomes Consortium project that actually showed that having low mass was not a predictor of negative health outcomes. So I think it was those series of papers that really forced this major shift in the operational definition of sarcopenia. So if we look at the effect of age on, say, muscle strength, the figure on the left is sort of showing the percent change in men and women, sorry, not men and women, in muscle mass and in muscle strength. So this black line is showing it from a cross-sectional study from the Health ABC study. It's showing the decline in mass between 20 and 85 years of age. But I want to note that the decline in muscle strength far exceeds it. I mean, it far exceeds the loss of mass, which either suggests that there's not a one-to-one relationship, right? That the loss of mass has a non-linear relationship with the loss of strength, or that there's a lot of other mechanisms going on. Now, the data on the right is also from the Health ABC study, but this is a longitudinal analysis. The women are in blue, the men are in orange, And this is actually from a subset of individuals who were between, I think, 70 and 79 at baseline. And they tracked them for 5 years. And in the subset of individuals that gained weight, they actually gained total body muscle mass. Actually, this was not total body muscle mass. This was quadriceps CSA, cross-sectional area. I don't remember if it was from a CT scan or an MRI, but it was definitely from a highly sensitive assessment technique. But despite gaining muscle mass, you still see that these individuals still exhibited a very large decline in muscle strength, which in my mind is pretty damning evidence, at least as it relates to the classical assumption of mass being the sole explanatory variable of the weakness. I was a muscle biologist by my training with my dissertation advisor. I'm not knocking the muscular system. I still think muscle is extremely important. So don't take my words out of context that way. I still am a huge proponent of skeletal muscle health and preserving a large amount of mass. But what I am trying to make the case is that there's other mechanisms that are driving this weakness in large part, and that to some extent we've been barking up the wrong tree when it comes to looking at the therapeutic interventions to promote muscle and motor function in older adults. And so I'm really going to focus on the neuromuscular mechanisms of muscle strength and weakness, with a heavy focus on the neuromechanisms. This was actually from the paper I mentioned that Todd and I did quite a few years ago, making the case for the strength not necessarily being explained by mass. And we were just sort of trying to highlight the various mechanisms that are driving the force output from the muscle. And just for simplicity, I think we can break it into, there's the brain. You've got the descending command coming from the supraspinal centers. We all know that the nervous system is an extremely complex cellular network of billions of cells and trillions of synapses. All of that has to functionally work together to generate a descending command to activate the muscle. And there is clear evidence that I'll show you in a bit that there's impairments in this descending command. You also have the discharge properties of these supraspinal connections, or this is just trying to say that it's a balance between excitatory and inhibitory inputs, as well as the intrinsic electrical properties of a neuron. And we're going to talk a little bit about that in a bit. Then we, of course, have the motor unit, the motor neuron, and the skeletal muscle fibers that it innervates. That's the sort of the final pathway that goes to the muscle activation. So you could have changes at both the supraspinal level, spinal level contributing to it. And then, of course, we have spinal reflexes that contribute to the neural control of muscle contraction. We've got reflexes that are required to integrate sort of the functional output, as well as the neuromuscular junction and potential transmission failure at the NMJ, which we'll show a little bit of data on in a second. So what's the evidence and what do we know? One of the things I want to point out, if we sort of look at the hierarchy of evidence, there's super strong evidence and there's weak evidence. When it comes to our evidence for neural mechanisms of weakness, we're mainly sitting somewhere down here in this sort of mid-level evidence. We don't have a whole lot of randomized controlled trials that are looking at the effects of various neurotherapeutic interventions for enhancing muscle function in older adults. We certainly don't have enough data for meta-analyses and systematic reviews. There's not even a whole lot of longitudinal cohort studies. They're mostly case control studies. So I point that out to point that there's still a lot of work to do in the neural mechanisms of weakness space. We're well behind where the field would be as it relates to sort of the skeletal muscle, regulatory control of muscle form and function with aging. This is just some preliminary data, and this is something we've not published yet, but I just show this to point out that what I'm calling quality, neuromuscular quality, is reduced in aging. And again, this is a small subset of older adults. I don't want this to be a definitive sort of take home, but this was in about 10 older adults and 10 younger adults. We tested their isokinetic leg extension strength, and we did an MRI of their quadriceps. We precisely calculate how much muscle volume they have in their thigh muscles, subtracting out fat. And we've simply expressed their voluntary muscle strength relative to their muscle mass. So we're kind of controlling for the mass component. And if we compare adult controls. I think these folks were on average about 30 years of age. They probably ranged from 22 to 50 or so. That's shown here in sort of the teal color. And in red, these are predominantly, depending on how you define it, sarcopenic or mobility limited or weak older adults. There's a bunch of these different operational definitions, but by all definitions, these individuals in red are very low functioning, weak older adults. The mean age was 86 years of age. We had one older adult here who was not weak. This particular individual had high physical function, and I point out that they also had preserved muscle quality. But what I want to point out is that these low functioning older adults clearly have very low levels of muscle quality. So there's more to it than just the mass going on. So the question that we always ask at sort of the really highest level is, do impairments in the nervous system's ability to generate maximal force contribute to sarcopenia? And here I'm kind of referring to sarcopenia by that most recent definition of weakness and or mobility limitations. I want to start with grip strength, because grip strength is where there's the strongest evidence of weakness being linked to disability and aging. And a lot of that's just because it's easy to do. It's quick to do. And it's been done in a lot of these epidemiological studies. So we've got a lot of evidence for it. But I'm going to argue that it's not a simple task. I always get irked when I read the papers that will refer to it as, oh, grip strength is a easy and simple task. Grip strength is actually an incredibly complicated physiological task. Our hands are extremely evolved as a functional unit. And to do a grip task, it takes 19 muscles within the hand, 20 muscles within the forearm, and all of these have to be sort of activated in a synergistic synergy is the word. You have to synergize them together to form the task. And the brain, or the homunculus, the motor homunculus, has massive representation for grip. And there is really nice evidence suggesting that impairments in neural activation are a major contributor to grip weakness. So this is actually not original data. This is some data that came from Minonoro Shinohara, who's now at Georgia Tech. I think this was probably when he was a PhD student from 25 years ago in Mark Latash's lab at Penn State. We just sort of reprinted this. This is along with Richard Carson, a paper we did just last year. But it's basically looking at what we call the multi-finger force deficit. And so this is not the exact, this is the device we have here that we use, but this is similar to what they have. But basically, you have a grip dynamometer that each individual digit can be measured from. So you can measure the grip force when you have somebody do a composite task, and then you can measure the grip force when they activate each digit individually, but you're still controlling for changes in biomechanical parameters. And what you observe is that even in young adults, when people have to perform it compositely, they have about a 30% less force output than if they do it with each individual digit, suggesting that that's an impairment when you have to do this synergistic composite task, an impairment in the nervous system's ability to generate force. But I want to point out that with the older adults, this force deficit is even larger. So it doesn't tell us why necessarily there's this neural impairment, but I think it makes a nice case to suggest that that grip strength in particular is heavily influenced by the nervous system, and that it's probably even more influenced by the nervous system in older adults. And so all of this is sort of going to suggest that the brain and the nervous system, both the central and peripheral nervous system, are likely playing a pretty heavy role in weakness as well as mobility limitations. And this is some data that I think this literally just got published 2 or 3 weeks ago. This is some data that we recently put out. I think it was a sample size of about 60 older adults, where we had T1 brain images, and we used a previously validated machine learning algorithm to basically calculate biological brain age. So everybody is sort of familiar with biological age from DNA methylation techniques or other telomere length kind of techniques. There are other approaches where you can use brain imaging and apply these machine learning algorithms to try to get an idea of whether somebody's brain is older or younger than their chronological years. So we basically calculated this sort of what we call brain predicted age or biological brain age. And then we looked at this sort of muscle strength to mobility relationship that people have reported for decades now, showing that there's a relationship between muscle strength and mobility. People who are weak tend to have worse mobility. And what we observed was that indeed that is the case, and it's really a strong case. So if we look at the relationship between strength and mobility on these two axes, for the older adults that had an older biological brain age, that relationship was significantly stronger than those with younger biological brain ages, suggesting that that sort of relationship between strength and mobility is being moderated by biological brain age. And so one of the things we are really interested in is the neural mechanisms. And so now I'm going to sort of pivot and go into a little bit more detail on what some of these neural mechanisms are. And I'm going to start with a couple of papers that we published first in 2019 as a brief report, and then the full report came out in 2021. We had 66 older adults in a study, and we had 20 young adults. The mean age of the older adults was 75 years. We then quantified the older adults' leg extension strength, their isokinetic leg extension strength at 60 degrees per second, and we expressed it relative to their body weight. And the reason we do this is that there's a number of papers that have shown that there are clinically meaningful cut points of weakness that are predictive of future development of mobility limitations. So for instance, men who have a value of 1.12 Newton meters per kilogram or women below 1.0 are at much higher risk of subsequently developing mobility limitations, as opposed to men and women who have values that are more in this 1.72 to 1.35. So we sort of applied adjectives to them to call these individuals over here weak, modestly weak, and not weak. I'm not referring to these individuals as strong necessarily. That may be a different cut point. We're just calling them not weak. And then we're calling the young adults all not weak. And just notice that the young adults are 3 times stronger than the weak older adults. We assessed a few different key outcomes from this study. The first one is what I'm calling neural activation. Some people refer to this as central activation, some as voluntary activation. And that's done using a combination of voluntary and electrically stimulated muscle contractions. And then we did magnetic brain stimulation on these individuals to get indices of corticospinal excitability, which I'll talk about in a second. And then in all of these analyses, we co-vary for biological sex as well as upper thigh lean mass to try to account for the influence of lean mass. But I want to just point out over here in this table, if we look at our not weak versus modestly weak versus weak group, there was no difference in appendicular lean mass to height squared or their lean thigh mass amounts. In fact, the weak groups actually had slightly higher, albeit non-significant levels. Again, going back to mass, certainly not being the full explanatory variable. So when I apologize, this is not the high resolution that I typically like to see, but that's okay. To assess neural activation, we have an individual sit in an isokinetic dynamometer, and we hook their leg in. This is a right-handed, or we're testing the right leg in this example. We normally do the non-dominant leg, so we flip back and forth. We've got them sitting in a dynamometer, their legs hooked in at 90 degrees angle, at knee angle, and we ask them to push out voluntarily as hard as they can. So they're given a maximal contraction, and that's what this little trace is showing here. This is torque. Somebody's pushing as hard as they can. We're giving them absolutely crazy strong verbal encouragement. People that have offices near us say that they think people are constantly going into labor in my lab because we're always yelling push, 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 push at the top of our lungs. Then while they're pushing out as hard as they can, we blast their muscle with direct electrical electrical stimulation. So before we've done this, we've already gone through and we've turned the current up until we get the maximum response from the muscle. And then we turn it up 15% above that. And then we ask them to push out as hard as they can. And then we put in 2 pulses 10 milliseconds apart. So it's 100 hertz double to basically blast the muscle. So if we see force go up at all, it tells us there's some sort of a suboptimal activation of the muscle. We then tell them to relax. We stimulate the muscle again with the same paradigm, and we calculate how much did force go up during the contraction relative to after the fact. We use an equation to basically calculate either voluntary activation, or the inverse of that would be voluntary inactivation. In this particular figure, I am reporting it as inactivation. The reason I report it in inactivation, if somebody's wondering if they've read our work and they think, why in the world do you sometimes call it activation versus inactivation? This was a figure that we did, and the journal would not let us scale things relative to the expected scale. It had to start at 0. And when we go from 0 to 100, if we go voluntary activation, these things start getting not easy to see. So we just flipped to the inverse so that it was a little bit more apparent. So when we compared our older adult subjects to our younger adult subjects, we observed that the young adult subjects, they had about 7 or 8% inactivation, and the older adults only had 10% inactivation. There was no statistical difference between the two. But obviously, you can see there's a lot of heterogeneity in our older adults. And that is something that I beat the drum on a lot, is that there's massive amounts of heterogeneity in aging. It's something I personally think we've not given due enough attention to over the years. And as somebody who works a lot with older adults on a daily basis, I always say we see some people who are 80 that are as healthy as a 60-year-old, and we see some people that are 65 that are doing worse than our 90-year-olds. So this is not overly surprising. What's interesting to us is that when we break these individuals into our not weak, modestly weak, and weak groups, this is where we see the difference. You start to suddenly see that our weak older adults are showing significantly more impairments in neural activation. We're talking about 15% inactivation. And assuming it's a linear relationship, that would argue that some sort of a neurotherapeutic approach could substantially enhance muscle strength in older adults. And in particular, I want to point out that a good proportion of both the modestly weak and the weak are showing impairments of 15 to 20, 25+%. But again, that doesn't tell us the mechanism. That just tells us that somewhere in the neural pathway, there's an impairment. So what are the neural mechanisms? So that's where we start getting into things such as, is there a change in the cortical or the supraspinal descending command? What about motor neuronal or spinal firing responsivity or neuromuscular junction transmission failure? So we assess all of those various things to try to get insight into that. So in the same group of subjects that I showed before, described before, we also did transcranial magnetic stimulation. For those who are not familiar with this, it's really simple, actually. We just basically apply a rapidly changing magnetic field to the motor homunculus section of the muscle we're studying. So in this case, the quadriceps. And it will elicit a motor evoked potential if we have the stimulation turned up sufficiently high. And we standardize that, of course, based on the motor threshold and things of that nature. And we turn it up for those who are in the space. We go 30% above active motor threshold for this particular study. And in this particular study, we measure motor evoked potential amplitude as well as silent period duration for indices of corticospinal excitability. And if there's anybody who does TMS, we've also done paired pulse TMS protocols, obviously, to get at more intracortical excitability measurements. And we can dive into that if somebody likes to. I've gotten to where in my talks, I tend to avoid it because it just gets a little too complicated for a general audience. So I'll stay at the single pulse TMS, but I'm happy to dive into that. Then we had them do contractions of 5, 20, and 40% intensities where we did the TMS, and we measured both the motor evoked potential and the silent period durations during this. And what I really want to point out is that particularly during the higher intensity contractions, we see substantially lower motor evoked potentials in our weak and our modestly weak older adults compared to our not weak older adults, as well as longer silent period durations, again, suggestive of corticospinal hypoexcitability, less excitability in the system. From this data, we certainly can't say it's the cortices versus the spinal cord. Based on our other data that I'm not showing, I would argue that it's probably both. We have data suggesting that it's both intracortical excitability is reduced, as well as data suggesting that motor neuronal excitability is reduced. And boy, that is a terrible resolution slide, but it's okay. I can describe it to you. We also took this data and we did multiple regression analyses where we basically put our brain stimulation derived physiological variables into a block. I'm calling that neural excitability. So there's two predictor variables in our neural block. We put fine lean mass into a block for muscle mass, and we look to see what's the relative contribution of neural excitability versus muscle mass and explaining the between subject weakness. And we observed that about 33% of the between subject weakness was being explained by excitability, and that was even slightly more than what we observed with lean mass. Now, I should note that there's 37% explained variance, so that could be other, neural measurements that we're not measuring here are not being reflected, as well as the ever obvious option of excitation contraction coupling that we did not assess. On a side note, this is eerily similar to some of the data that we see with disuse. In addition to the aging studies for years, we've done various disuse models to try to parse out the neuromuscular mechanisms of weakness. And in this particular study, this was a J Neurophys study from 2014. We took about 45 perfectly healthy young adults and we cast immobilized their arm for, I think it was 4 weeks. Yeah, it's just 4 weeks. So it's a long period of time to be in a cast. And that resulted in about a 50% reduction in muscle strength. About half, and that's shown here. This is shown in this line right here. And I should note these people bounce back. They're young, healthy people. They bounce back pretty quick. A control group that we just tested at various time points are shown here, and you don't really see a change, as we would hope. This middle group here is a group that, Jill, this is going to sound eerily familiar. I was doing some of this work as my dissertation, that did mental imagery training. So these individuals came into the lab 5 days a week, and they imagined doing 50 maximal forearm flexion contractions. That's all they did. They did not do anything else except come in and they thought about doing contractions from a kinesthetic or a first person perspective. And then before and after this, we assess voluntary activation. Now here I'm expressing it as activation, not inactivation. But what you'll see is that this cast immobilization group had a plummet in their ability for neural activation, whereas this group here was the imagery group, attenuated it substantially. But what I wanted to point out is that we observed similar neurophysiologic findings. In particular, this is just showing the silent period duration. That group that we were able to attenuate that sort of strength loss and the neural activation impairment, it attenuated the prolongation of the silent period. So I don't think it's a model of aging at all, but it is eerily similar to some of the findings that we observe with aging. So I sort of said before, I think it's probably both the reduction in excitability of both the upper motor neurons as well as the lower motor neurons. This was some data that just came out in the past 4 or 5 months, not from our group, but from a different group, looking at an intrinsic assessment or an indice of motor neuronal excitability with aging. And the short answer is that they observed reductions in this motor neuronal excitability index. Specifically, this is an approach that looks at single motor unit firing patterns during a triangular task contraction. And you basically look to see when does a motor unit kick on and kick off. And they typically kick off at a lower recruitment threshold because the contraction itself is activating persistent inward currents. And that's sort of what they're referring to as this delta F. It's the number of hertz or the peaks per second that it kicks off at a different level. And they observed that both for the soleus and the tibialis anterior, the older adults had a lower delta F value indicative of intrinsic motor neuronal hypoexcitability. This is some work, we just got an R01 last year that I'm not really presenting any data on here yet, but that's barking up this tree heavily. And in that work, we're going to expand and kind of go back to that prior thing to look to see whether or not we see really weak older adults having extremely low measures of intrinsic excitability. And we're also doing some animal work with colleagues at Ohio State, Dave Arnold, as well as Sharif El-Bassiouni at Wright State, looking at more cell and molecular kind of mechanisms. This is some work that we've not published yet, but that we're hoping to send out soon. That's sort of along the same line, looking at the motor neuron. And this is showing that slowed motor unit firing contributes to leg extensor weakness. And for a very long time, dating back at least 30 years, there have been reports showing, with Gary Kamen's work being the first that comes to my mind, showing that older adults have slowed motor unit firing rates compared to younger controls. But to our knowledge, nobody has actually tried to map this to clinically meaningful weakness. And if I kind of go back to that hierarchy of evidence, I would, I guess, advocate that, comparing older adults to young adults is sort of like level of evidence 1, and that the next level of evidence is mapping it to a phenotype, and then such as weakness. And then from there, it's sort of moving it into a longitudinal cohort kind of study, and then an interventional perturbation kind of paradigm. And so this particular experiment was trying to map it to the phenotype. So we had older adults do a variety of different leg extensor tasks while we were using some of these more recently developed surface decomposition EMG techniques that try to derive motor unit action potential firing rates. We plotted the motor unit firing rate versus the recruitment threshold to identify the y-intercept, because there is, I've never fully understood why others don't do this as much, but there's clearly a relationship between firing rate and recruitment threshold. So the reason we're using this y-intercept as our primary outcome is just trying to control for that. So that's what I'm referring to over here as the normalized firing rate. But what I want to point out is that just similar to the prior literature, we did observe that particularly at higher intensity contractions, older adults had a slightly lower firing rate. It was about 2.5 hertz. And that when we look at the not weak versus the weak older adults, we see another 2.5 or 3 hertz difference there. Again, I believe suggesting that weak older adults have lower degrees of firing rates. And interestingly, these were associated with various indices of both neuromuscular quality, that's sort of muscle strength relative to muscle size, as well as outcomes of physical function, such as chair rise time and stair climb power. We are working with the El-Bassiouni lab at Wright State to actually model that data to try to get an idea of how much of a reduction in force would a 3 hertz firing rate be. We don't think it's linear because of the it probably largely affecting the larger threshold firing rates, but we don't have that results back yet. But we are looking at trying to do some computer modeling to get an idea of what kind of an actual expected force reduction that would yield. And in addition to sort of changes in motor unit firing patterns, this is sort of data showing that there's also a loss in motor unit numbers or a loss in motor units. This is just cross-sectional data. On the left, this is actually, I think, data probably from the 70s. I think it looks like the data probably from Alan McComas's group from his classic textbook. I found it because I was looking for a better figure in a more recent review article. But I think this is probably original data from Alan McComas's group. But what you'll see is that, at least in these cadaveric studies, there's a relatively, there's a lot of heterogeneity, but if you sort of do a linear regression line at the younger ages, it seems to be fairly stable. And then after a certain age, there's a gradual reduction in motor unit numbers. Now, I doubt personally that it's truly a linear response. I would probably believe it's much more of a cyclical response, that some of these losses are probably due to various health events, but when you just do these kind of scatter plots, you don't model that. And then on the right, this is some data from our group from about 10 years ago, looking at the relationship between strength and motor unit number. And I think the take home here is that we see absolutely no relationship when we look at this sort of relationship in young adults, as we would expect, but that in older adults, we see some degree of a relationship between a fewer estimated number of motor units being linked to reduced muscle strength. And this was specifically with pinch grip. The last piece of original evidence I think that I'm presenting is some work looking at the neuromuscular junction. This is some work that we've not published yet. We'll be presenting this in a few weeks at the International Conference for Frailty and Sarcopenia Research. And in this particular, this is actually, I think I sort of showed this figure earlier. We only had 8 older adults. This is a proof of concept study. We had 8 young adults, 29 years of age, 10 older adults, 86 years of age. We assessed their leg muscle strength, quadriceps MRI, and then we performed stimulated single fiber EMG. So what this is a complicated technique. I do not do the technique myself. My colleague Dave Arnold from OSU, who is a neuromuscular medicine electromyography subspecialist, he comes down, he's about an hour away and performs the technique. He basically, I'm trying to sort of illustrate this here. He takes a very focal recording electrode and sticks it into the vastus lateralis. Then he takes a stimulating probe. First, he takes a handheld probe and he identifies where the twigs of the motor point are. And once he sees where the twigs are, he takes 2 needles that are, and this is a needle EMG electrode as well, but he takes 2 stimulating electrodes and he puts them on each side of them where he thinks each side of the twigs are. And of course, this is our subjects say they feel like a pin cushion when we're doing this, because it's not as simple as 1 needle here and 2 needles here. There's a bit of the having to move it around to optimize the recordings. But effectively what we end up with is we have 2 stimulating electrodes, an anode and a cathode, and a recording electrode. And this recording electrode has a very, very focal recording, so we can actually get a single muscle fiber action potential. And so we'll apply a stimulus from these stimulating electrodes, and then we look to see, do we get a time-locked action potential? And up here in the top portion, this is what we would expect in a perfectly normal individual, that every time you stimulate, you get the action potential, and that they're time-locked, and that if you superimpose them, you get these perfectly time-locked action potentials. What we'll see in an abnormal signal is a couple of things. One is you see that they're not as time-locked. It's what they call jitter. And you're seeing that here. If you superimpose all these, you'll see how they're not as time-locked. There's some variability in the transmission. But we also see blocking. And what blocking refers to, in a very simple term, is that's an action potential that is failing to propagate to the muscle. It literally is not propagating and resulting in an action potential in the skeletal muscle. And so this is the data. If we, again, I wanted to start over here just showing muscle quality. When we look at the sort of the relative quality of the muscle, the voluntary strength versus the muscle mass, we took these older adults and we broke them into weakness groups again. So sort of mainly focus on this red group relative to the healthy controls. This was the one outlier older adult who is 89 years of age, who is a phenomenal ager. I mean, she's just doing phenomenally well in every aspect. We selectively tried to recruit people who we thought would be very low functioning based on age and self-report, but in reality, her physical function was really solid. Her gait speed was pushing 1.2 meters per second. But these lower functioning individuals had lower muscle quality. They had substantially higher jitter, as you can see. But what I really want to point out is they had substantially higher blocking. Some of these older adults were showing the action potentials. Now, again, this is in a handful of fibers, or in one tiny location. We can't generalize it to the whole muscle, but for this one selected spot, we were seeing 15 to upwards of 30% of their action potentials that are being generated by the nerve failing to propagate to the skeletal muscle. And as one would probably expect, that had an association with physical function. Both jitter and blocking had an association with impaired muscle quality. So again, what I want to just quickly now talk about are what are the research/clinical gaps and recommendations for future work. Well, I sort of said that most of the work that has been done to date is sort of sitting down here in these cross-sectional studies and these sort of case control studies. I would put our work mostly in this case control, where we're trying to compare phenotypically described older adults to a different phenotype. But I think there's a real need to do more longitudinal studies as well as clinical trials. And just as a primer, this is all very tenuous data. I would not put any of the data that I'm showing here into high rigor. But there are, if you dive into the literature, you can find all sorts of very intriguing findings suggesting that there could be neurotherapeutics to target weakness. So for instance, transcranial direct current stimulation to increase cortical excitability has been shown to improve hand dexterity in older adults, as well as fatigue resistance. We actually did this fatigue resistance experiment. Well, I'm not showing the data, but we basically found that if we applied anodal TDCS to the primary motor cortex, we could enhance the time to task failure by upwards of 15% in older adults. Caffeine, another well-known central nervous system stimulant, has been shown to have fairly modest improvements in various parameters of motor function. And somebody may look at that and think 5–10% doesn't sound that great, but when you look at what the anabolic therapies have done, this is actually pretty impressive. And then, of course, you've got the ADHD meds as well as L-DOPA have shown even more robust increases. L-DOPA, and again, this is a very small study. I forget exactly how many people. It was only dozens of people, not a large clinical trial, and it was not at all. You have to dig into this one paper to find this. That was one focused more on depression. But they just haphazardly happened, not haphazard, they happened to measure single task gait speed and dual task gait speed in this study. And they reported that administration of 3 weeks of L-DOPA enhanced gait speed by 15 to upwards of 30% for a dual task, which is a very robust increase. And I think there are some scientists at UPitt that are actually currently doing a sort of a proof of concept study looking at the effects of L-DOPA on enhancing mobility and aging. So I suspect that in the next few years, we may see a little bit more of that. So in conclusion, neural mechanisms contribute in part to age-related weakness and mobility limitations. It's sort of going back to the my initial statement that the muscles are puppets of the nervous system, and it likely explains why the anabolics have not had the desired effects. These neural mechanisms are multifactorial. They include arguably supraspinal changes, potentially linked to both reduced cortical excitability, as well as dopaminergic dysfunction and arousal. The spinal changes are potentially linked to loss of functioning motor units, reduced intrinsic motor neuron excitability, as well as slowed firing rates. And then I think I've also provided a little bit of data to sort of whet appetites about NMJ transmission failure. Again, I would argue none of this is super strong levels of evidence. It's case control studies. They're not longitudinal aging studies, and that we need a lot more information. And then, of course, trials to really push this field forward, which is my sort of future work, is to determine the clinical relevance of these physiological changes by mapping these putative mechanisms to negative health phenotypes, and then, of course, examining the effectiveness of various neurotherapeutic approaches on enhancing function. And I don't just mean, I am an exercise physiologist by my training. I don't refer to this necessarily as it relates to drugs only. I'm a huge advocate of physical activity for maintaining these neural pathways and activation patterns, as well as, there's all kinds of evidence for various nutritional strategies to promote mitochondrial health and things of that nature that we know are linked to muscle function. So I'll stop there. I will just quickly acknowledge some of my trainees and collaborators, both here at Ohio University, as well as my external collaborators from the US and other places. And I'd be happy to take questions.
Brian, thank you very much. That was a great presentation. If people want to unmute and clap, give them a clap. I think we have time for a few questions. If you want to stop sharing your screen, then we can see the gallery and people can jump in. I know we have a couple questions on chat that I can look at. One question that I have is, you're seeing a decrease in the number of motor units with aging. Are they primarily type 2? Going to the type 2 muscle?
So probably so. I think the short answer to that's based on the data we've done. We can't necessarily conclude that because we're using non-invasive techniques, but I think it's fairly safe to say, based on some of the cadaveric studies, that they're the larger threshold fibers.
Okay. So question from James Smith. Let's say two people are 80 years old, healthy, and had similar resistance training programs throughout their life. One person did a lot of brain games, challenges on a consistent basis, crossword, Sudoku, that type thing, while the other person did not. What are the what are your thoughts on the potential neuro differences in the neuromuscular strength? Does it play a role?
Yeah, that's a question I don't fully know, to be honest with you. I work very closely with a geriatric neuropsychologist, Julie Sewer here at OU, and these are the kind of questions I ask her. And she'll frequently tell me, maybe, maybe not. There's a lot of potential, genetic factors that could influence it, that she'll typically lead me to the thing of the two things we know that seems to enhance sort of the maintenance of cognition, at least, which is the one thing we can pretty certainty know is a neurological system. It's not got the muscle in it. Is aerobic activity, good vascular health, and good nutrition. So a lot of it relates to, I think, the vascular health there. Certainly there's a lot of work linking, motor and cognitive function. Joe Verhese and others have done work sort of suggesting that there's some sort of an interdependence between age-related declines in cognitive function and motor function. There's the question of which comes first, the chicken or the egg. But certainly when we do dual task paradigms, when you stress the cognitive system, you see impairments in motor function. So I think the real question comes down to whether or not crossword puzzles and Sudoku work. I hope it's chess. I play chess with a buddy every Sunday for 2 hours. So I think we all have our hopes on some sort of a cognitive thing working, but I honestly don't know. I've not read enough in the literature there.
Great. So the use of creatine supplementation by athletes, whoops, it just moved up on me, by athletes to increase muscle mass and hence performance has been debatable. Given your expertise in this field, what do you think about this? Anything known about creatine supplementation?
Yeah, to be totally honest with you, I think you guys probably know way more about that than me. It's been 20 years since I probably stayed abreast of that literature. And so I'm a little skeptical to dive into it. Creatine supplementation is not something the older adult community is doing, so I've sort of pivoted away from it. So I really could not, with an honest attempt, tell you what I know about the creatine supplementation today.
Okay. From the question from Jopna, what mechanisms are responsible for reduced motor neuron excitability and how do you test those?
Okay, so that's more in my wheelhouse. I can address that. I'm more comfortable in that space. So the mechanisms can be multifactorial. There's a lot of things that will influence the excitability of a motor neuron. It could be the sodium-potassium pump channels. At a really high level, probably the most robust thing is the physical diameter at the axon hillock. People have heard of the size principle. So a small diameter motor neuron is going to reach threshold and be more excitable than a large diameter motor neuron because of Ohm's law. Now, that's not something that we necessarily expect to dramatically change with age. That's what's sort of determining the excitability of that within us and why we have that sort of orderly recruitment. Although, atrophy of a neuron would affect that to some extent, but there's a lot of other things, both intrinsic to the motor neuron, like those sodium-potassium channels, as well as mechanisms external to the motor neuron, such as the presynaptic inputs coming from other sensory inputs that will determine the excitability. In animals, we can test excitability much more direct. You can actually excise the neuron, you can measure the voltage, you can inject current, and you can measure input-output relationships. I don't personally do those techniques, but I do have collaborators that we're doing those as we speak, really trying to do a deep characterization of motor neuronal excitability with aging and mapping that to those exact, those pumps and other things of that nature, and particularly the persistent inward currents. Now, in humans, how do we go about the testing motor neuron excitability? There are a few different ways. Some people will use the H reflex, which is where you basically have an EMG electrode over a muscle, you electrically stimulate a peripheral nerve, and you get a sort of a spinal reflex loop. I argue that that's a very, very, very indirect assessment, that there's about 5 potential sites within that reflex loop that could actually change what you observe, with the motor neuronal excitability only being one of them. such as the amount of neurotransmitter being released from the sensory neuron. So if you have that go up or down, you'll get a change in your reflex, even though that has nothing to do with the motor neuron. So I don't use that much anymore. What used to be my preferred approach, which we've only published in one study at cervicomedullary evoked potentials, where we put electrodes on a muscle and we either magnetically or electrically stimulate the cervicomedullary junction. And we've done it both ways, predominantly with electrical stimulation. And so you get a single descending command from the motor, from the upper motor neuron communicating to the lower motor neuron. There's not a lot of groups that have done that. Janet Taylor and Simon Gandivia's group out of Australia, or if somebody's interested in it, are the ones that have done it the most. The reason we don't use it much is that it hurts like crazy. I mean, and I'm talking, it hurts. I have volunteered myself for Jill's studies back in the day. I've done muscle biopsies on myself. I've stimulated everything. That is the only thing that we have done that I really do not like. It's very uncomfortable. I have never done it. I always say I will not do something to an older adult that I am not willing to do to myself without any hesitation. So we've never applied it to aging studies because of that. It really gives a pretty sharp whiplash effect. And I'd be worried about doing it in an older adult population, but it is a great way to get a composite measure of the motor neuron pool. The third way that we're barking up aggressively right now in humans is this delta F technique. It's called the paired motor unit analysis technique. It was developed about 20 years ago. And it's taken off. It's kind of complicated to do, though. You have to get a lot of motor unit action potentials during a triangular task, and look when they kick off and kick on. So it's a bit of a, it's a lonely task to do the analysis. And we're working with Chris Thompson at Temple for that analysis. We've developed a relationship with Chris where we collect the data here. And I think it takes deep expertise to do the analysis, and we just have a pipeline where we send it to Chris. So we've not published that work yet, but that is some of the work we're working on.
Thank you very much. I think we need to have you over again just for another seminar on what you just talked about.
Deeper dive.
Yeah. So we're over time. Brian, thank you very much for your time. If people want to unmute and give Dr. Clark a hand. Thank you very much. This was fascinating.
Thank you so much. Wonderful.
Thanks very much.
Jill, if I can say, I see there's one question relating to non-responders to exercise. We literally just published a paper on that. If you look up my name it was in experimental gerontology, and we specifically tried to parse out the mechanisms of responsivity to exercise. So if whoever's interested in that, they can take a deep dive there.
That's great. Thank you.
Okay, thanks everybody.
Yep.
Thank you.
Bye. Have a good night.
Thank you.